Unit 5, page 5. We're going to be talking about HIV. How does HIV cause someone to get sick? Do now. Let's review. What's a pathogen? What is an antigen? What is an antibody? Pause the video here to answer these three questions. When you're ready to begin, hit play. A pathogen is a microbe that causes disease. An antigen is the protein receptor on the surface of the pathogen. An antibody is produced by our white blood cells in response to an antigen. It marks the pathogen to be destroyed. We're looking here at a picture of HIV. So the virus structure, and we can see here kind of the cartoon image, shows that we have in the middle RNA, so we have a nucleic acid or genetic material on the inside. We have something called reverse transcriptase, which is an enzyme located on the inside as well. And then we have a membrane surrounding the virus with receptors on the outside. Now these receptors, remember, are called antigens. Viruses are neither living nor non-living. Viruses contain genetic information in the form of DNA or RNA. And viruses use receptors to enter cells. This particular virus, HIV, again uses RNA. So how does HIV infect your cells? Well, it does this in many steps. So the first step is binding and fusion. So HIV binds to two receptors on the cell membrane of the helper T cell, otherwise known as the CD4 cell. So there's two receptors located on the outside, one called CD4 receptor, and the other one is either CCR5 receptor or a CXCR4 receptor. So again, in order for HIV to infect the helper T cells, it needs to bind to the correct shaped receptor. After the binding, we use an enzyme known as reverse transcriptase. HIV's enzyme converts the viral RNA into DNA. Then integration. We're going to have the viral enzymes are going to aid the virus so that it can add its DNA into our DNA. So if you look here, the green represents the host DNA and the yellow represents the HIV DNA. So now you can see that this cell nucleus contains both viral DNA as well as host DNA. The next step is transcription. So we're going to have this DNA is then converted into viral RNA to make more virus. That viral RNA is then assembled near the cell membrane to make the proteins and the new virus particles. And then the last step, we have budding. So newly assembled viruses pinch off your cell membranes to make its own cell membrane. So we're going to look here at a picture of what this actually looks like. So again, you can see here the picture of all the virus particles budding off of the cell membrane. Now eventually, this cell will die because the virus will be ripping off all of its cell membrane. So HIV's GP120 antigen is going to target the cells because of the CD4 receptor. So basically, HIV's antigen targets our receptors. HIV takes control of the T cells that normally activate the rest of the immune response. So remember, T cells activate both killer T cells as well as B cells, which make the antibodies. Therefore, people with HIV/AIDS are susceptible to what we call opportunistic pathogens, things like pneumonia, influenza, thrush, which is a fungal infection. So people are going to have a compromised immune system, which means it's a weakened immune system, and going to get other diseases that will eventually make them sick. How do we test for HIV? Well, there's two different types of tests. One is the antibody test against HIV. This takes anywhere, though, from 2 to 8 weeks for you to develop antibodies against HIV. If you get a negative result in less than 3 months during possible exposure, you should be tested again because there is a possibility that you do have the virus within 3 months of contact and you're just not showing it. And then there's the RNA test. This looks for the viral genetic information directly, and it's a 9 to 11 days after you've been infected that it'll show up in your bloodstream. It's very expensive and it's offered by few states. Most states offer the antibody test. But like I said, it's not good if it's within the 3 months of you being exposed. For more information, you can look at hivtest.org or even call the CDC, which is the Center for Disease Control. Here's an example of what the HIV test will look like. So you can see here, this here would be a positive test. So you can see the control strip compared to the test strip, and over here you would see a negative test. So the control strip, and then nothing shows up for the test. Summary questions. Use your notes and knowledge of biology to answer the summary questions. These will be checked by your teacher, and we're not going to review them in the video. So remember, use your notes.